Good morning, students. We will continue the topic protein metabolism, competition number 5.4, and today's topic is transport of ammonia. Now, in the previous competency, we have studied transamination, deamination, transdeamination, and during the process of transamination, what we have studied is free ammonia is formed in various tissues. So if this free ammonia remains in the tissues, it may enter into the blood and it may goes to liver and it may damage to the various organs. So what we have studied in the previous slide, in all tissues oxidative deamination except oxidative deamination by glutamate dehydrogenase and non-oxidative deamination. This will lead to formation of free ammonia, whereas transdeamination leads to formation of free ammonia in the liver. Ammonia is also formed in alimentary canal by bacterial putrefaction action, and small amount of ammonia is also formed by the catabolism of nitrogen containing compounds like purines and pyrimidines in the various tissues. So these are the various sources of ammonia in the body. Now we will see how ammonia from muscles is transported to the liver. So in the muscles, when ammonia is formed due to any reason, maybe because of oxidative deamination, maybe because of transdeamination, or due to catabolism of nitrogen containing compounds. Now this ammonia combines with pyruvate to form alanine. Now this alanine is then transported to the liver via blood. In the liver, it undergoes deamination, that is removal of amino group, and free ammonia is released in the liver and pyruvate is converted, reformed. So the ammonia which is formed in the muscle is transported in the form of alanine to the liver. So sometimes they may be asked in MCQ, the transport form of ammonia from muscle to liver is, so answer is alanine, because ammonia is trapped in the form of alanine. Now how the free ammonia from brain and other tissues are transported to the liver. So in brain and other tissues, two amino acids are important. First is glutamic acid. This glutamic acid combines with ammonia to form glutamine. This glutamine goes to liver. In the liver, the enzyme glutaminase is present, which act on glutamine and convert that glutamine into glutamic acid and free ammonia. So in this way, the free ammonia which is formed in brain and tissue is transported to the liver in the form of glutamine. And next is aspartic amino acid. So here again, the ammonia which is formed in the liver and brain combines with aspartic acid to form asparagine. The enzyme present in these tissues is asparagine synthetase. Now this asparagine is transported to the liver. In the liver, enzyme asparaginase is present, which acts on asparagine and removes the ammonia from asparagine, free ammonia, and aspartic acid is resynthesized. So in this way, the form of ammonia which is transported from brain and other tissues to liver is in the form of glutamine and asparagine. So form of ammonia which is transported from muscle to liver is alanine. So the same reaction, glutamic acid combines with ammonia to form glutamine in the liver. Glutaminase acts on glutamine and releases the free ammonia. The enzyme glutamine synthetase is present in the tissue, whereas the enzyme glutaminase is present in the liver. Again, same, aspartic acid combines with ammonia to form aspargine. The enzyme aspargine synthetase. Now this aspargine undergoes breakdown in liver by enzyme asparginase to form free ammonia and aspartic acid. So aspargine synthetase is present in tissues, whereas asparginase is present in the liver. After studying the transport of ammonia, we will see in short about ammonia toxicity. Why free ammonia is toxic to the central nervous system. We will see the mechanism. The first mechanism is, if free ammonia is present in the cell, it will lead to energy deficiency of the cell. Now what is the mechanism? If free ammonia is present, it combines with alpha ketoglutarate to form glutamate. So there is deficiency of alpha ketoglutarate in the body. If more free ammonia is present in the cell, then more and more alpha ketoglutarate is converted to glutamic acid, and it leads to deficiency of glutamic acid, alpha ketoglutarate. And this alpha ketoglutarate is a intermediate of TCA cycle. That means this ammonia combines with the alpha ketoglutarate, which is intermediate of TCA cycle, and hence the TCA cycle cannot proceed further. That alpha ketoglutarate actually has to convert into next compound in the TCA cycle, but that alpha ketoglutarate of the TCA cycle combines with ammonia to form glutamate, and that TCA cycle will not be completed. And all of us know TCA cycle is the major cycle which provides energy for our body. So in this way it leads to deficiency of energy. Second, when more ammonia is present, the glutamic acid is converted into glutamine. That means alpha ketoglutarate combines with one ammonia to form glutamate, and that glutamate again combines with another molecule of free ammonia to form glutamine. Now this leads to deficiency of glutamic acid, and all of us know that a neurotransmitter called GABA, gamma aminobutyric acid, is formed from glutamic acid. When there is deficiency of glutamate or glutamic acid, there is deficiency of GABA, and it leads to imbalance of neurotransmitter. Third mechanism is damage to the cell. We have seen that more and more glutamine is formed. So this glutamine get deposited inside the cells, and it will damage the brain cell and central nervous system.